Well, hello and welcome back to part two of the lecture over chapter 23. And I don't know if I'll finish the chapter in this section. I may have to do a part three, I'm not sure, because there's really a lot of things to cover in chapter 23, and it takes careful explanation as well, because I want to make sure that you understand everything that's going on. And also, as always, I'd like to mention that if you have additional questions and things after you've listened through the lecture, just let me know, send me an email, and I can answer questions and things, and I'm happy to do that. And so anyway, let's get started then. And I think we've finished in part one. I think I finished part one on slide 47. So I'm going to start there today. So here then in slide 47, we see that again, we're talking about the Hardy-Weinberg equation. And remember, the Hardy-Weinberg equation describes a situation in which there's no evolution happening in a population or at a locus. And so the Hardy-Weinberg equation can actually be used to test whether evolution is occurring in a population. And it can be used to determine the percentage of the population carrying a certain allele as well. So there's two different things that the Hardy-Weinberg equation can be used for. And looking at an example then, so we can assume that the locus that causes phenylketonuria, that's a human disorder. So phenylketonuria, or PKU for short. Sometimes they call it PKU syndrome. But we can assume that the locus that causes PKU is in Hardy-Weinberg equilibrium because, number one, the PKU mutation rate is very low, and mate selection is pretty much random with respect to whether or not someone is a carrier for a PKU allele, because most people that are carriers for PKU allele don't even know they're carriers, and so mating with respect to that allele is pretty much random in the population. Also, going through the rest of the criteria for Hardy-Weinberg equilibrium, natural selection can only act on rare homozygous individuals. Since the allele that causes PKU is very rare in the human population, that means that being homozygous for it would be extremely rare. And natural selection can only act on those very extremely rare homozygous individuals who don't follow the dietary restrictions. Because if you follow dietary restrictions, then you can live with PKU and you can live a normal life and everything's fine. So it's a rare event that natural selection can actually act against someone in that with that condition and who's not following the dietary restrictions. Also, the human population is very large and getting larger every day. And basically, migration in or out of a local population has no effect because pretty much every other population has very similar allele frequencies. So basically, Hardy-Weinberg equilibrium is probably present for the PKU allele. Now, so the occurrence of PKU, the PKU allele, the occurrence of the PKU condition is actually 1 per 10,000 births. And in order to have, so the PKU allele is recessive again. So in order to have the PKU condition, you've got to be homozygous for the PKU allele. You've got to have 2 of those PKU alleles. So in other words, you've got to be Q squared in the Hardy-Weinberg equation. And so basically, if PKU, so then we know that the proportion of the population that has the PKU condition and therefore is homozygous is 1 per 10,000 births. That means that, in other words, it's Q squared. And 1 out of 10,000 is 0.0001. And so from this, then, the square root of this 0.0001 is 0.01. So then we know from there, basically, we know the proportion of Q in the population. And from the proportion of Q in the population, we can easily calculate P in the population, because everything has to add up to 1. So P would actually equal 1 minus Q. So it's simple algebraic manipulation of the equation P plus Q equals 1. So if P plus Q equals 1, that means if you solve for P, then P equals 1 minus Q. And we already know that Q is, from up here, we already know that Q is 0.01. So then 1 minus 0.01 actually gives you 0.99. So therefore, the frequency of P in the population is 0.99. And so the frequency of the carriers, now the carriers are the heterozygotes, okay? So once we know what P is and what Q is, then simple calculation will give you, so 2 times what P is, which is 0.99, we've already found, times what Q is, which is 0.01, we've already seen that from up here. So basically then 2 times, if you put this into a calculator, 2 times 0.99 times 0.01 gives you 0.0198. And so there you go. So that's how you can actually use the Hardy-Weinberg equation then to actually calculate the proportion of homozygous P in the population, homozygous Q in the population, and also you can calculate then from there those in the population that are heterozygotes. And the heterozygotes are called carriers because they have the recessive allele, but they're not expressing the recessive allele. So they're called a carrier then. Just like a carrier for hemophilia or a carrier for color blindness or whatever. So approximately then, here then, this is approximately 2%. If you convert this to a percentage, it's approximately 2%. So approximately 2% of the US population is a carrier for the PKU allele. So that's a really good example then of how to use the Hardy-Weinberg equation to figure out the proportion of different types of individuals in a population for some certain gene or some certain locus. So now let's go on then. There are 3 major factors that alter or change allele frequencies in a population and bring about most evolutionary change. So most evolutionary change and therefore most changes in allele frequencies in populations is brought about by either natural selection, which we've already covered, or genetic drift, which we're going to cover, or gene flow, which we'll cover as well here in the next few slides. And so here's a little video that you can watch on genetic drift, which is kind of interesting to see. So you can watch that on your own time, and if any questions come up, let me know. And then here's another animation on causes of evolutionary change. You can watch that on your own time as well. So basically then, going over a little bit more natural selection. So natural selection then basically results in alleles being passed on to the next generation in proportions that differ from those in the present generation. So if there's natural selection acting on certain alleles, then it's going to change the proportion of those alleles in the next generation, and probably a number of future generations. So for example, let's say an allele that confers resistance to DDT in fruit flies increases the frequency. Now, DDT, I should give you a little bit of background, because DDT was first developed in the late 30s, and it first started being used. It's an insecticide, first developed in the 1930s, late 1930s, and it was first used in, I think, 1943 to control insects. But by the early 50s, insects had already developed resistance to DDT, and we basically phased out DDT sometime in early 70s, and a lot of other countries followed suit as well. But then back in 2006, since it was several decades since DDT had been used, that the World Health Organization actually authorized the reuse of DDT in limited amounts. But anyway, so it's one of the most well-known insecticides from history anyway. So that's what DDT is. So an allele that confers resistance to DDT in fruit flies increased in frequency after DDT was used widely in agriculture, and that makes sense. Because if, once they started using DDT in agriculture, then any fly that had an allele that gave it resistance to DDT, that fly was going to be at a very great advantage over others that weren't resistant to DDT. And so the flies that had this allele that was causing them to be resistant to DDT were able to survive at a much higher frequency than flies that didn't have an allele that gave them resistance to DDT. So natural selection then, in other words, in that case, natural selection then can cause adaptive evolution. Adaptive evolution then is the process in which traits that enhance survival or enhance reproduction increase in frequency over time. And pretty much all evolution would be adaptive evolution then, because adaptive evolution just means you're increasing the ability of a population to survive and to reproduce in a certain under certain environmental conditions. So adaptive evolution then. Now we're going to talk about genetic drift then. Genetic drift is kind of an interesting phenomenon. And drift is like a random movement, right? So genetic drift is actually a random shift in allele frequencies in a population, and we'll see this now. So when you are looking at a population and you take a sample of individuals from that population, you're actually taking a group of individuals from that population, and you're testing them for whatever it is you're testing for. So they're talking about samples here, and that's what they mean when they talk about samples here. What they mean is, if you're doing sample in the sense that it's just a small proportion of a population. So, but the smaller a sample of individuals from a population, the greater the chance of random deviation from a predicted result. And we'll see examples of this so that it'll make it more clear. So genetic drift then is basically how allele frequencies can fluctuate unpredictably, in other words, randomly, from one generation to the next. And genetic drift tends to reduce genetic variation through just a random loss of alleles. And we'll see that we're gonna go through a good example here, and we'll explain what they're talking about in terms of genetic drift and samples and fluctuation of allele frequencies and things like that. So picture then we're going to go back to the flower example again. These could be snapdragons or 4 o'clocks or whatever. But in this population then, There are some red, there are some pink, and there's some white as well. And so the frequency of the red allele is 0.7, and the frequency of the proportion then of the white allele, or frequency, is 0.3. But let's say an herbivore, only 5 of these plants leave offspring, and let's say an herbivore, eats 5 of these plants, but the other 5 are left to leave offspring. So basically, then just 5 of the plants, just a small number, just 5 of the plants actually leave offspring, and just by random chance, the white one and the yellow one, and just by random chance, fewer red ones are left to leave offspring, but more white ones are left to leave offspring. And so that means in generation 2, then the frequency, the allele frequencies have changed a little bit, just at random. We're assuming this happened at random. And then in the next generation, only 2 plants, maybe only 2 plants were able to leave offspring. And so this is a small proportion of the population. In other words, a small sample of the population. Just a small sample of the population were able to leave offspring for whatever reason, and for whatever reason, just at random, the small proportion of the population that was able to leave offspring just happened to be red. And so in this case, then, in generation 3, all of the offspring actually have the red allele, and none of them have the white allele. So basically, a proportion of 1 or a percentage of 100% have the red allele and none have the white allele. So you can see that if you've got a small population size, and it's cut down even smaller, if you take a small sample from that population, if you take at random a small sample from that population, then at random you're going to get some pretty large shifts in the proportion of the individuals that have certain alleles in the population. So you can see, from generation 1 to generation 2, there was a pretty substantial shift, and from generation 2 to generation 3, again, there was a pretty substantial shift. And so this is a good example then that explains what we were talking about back here. So if there's a small sample, a small number of individuals from a population that are only able to leave offspring, then there's going to be a greater chance of some sort of just random deviation from a predicted result, or it could be a random deviation from one generation from another. So then this is allele frequencies fluctuating unpredictably from one generation to the next. So that example then explains what they're doing here with the definition of genetic drift. And also, genetic drift tends to reduce genetic variation, because if you take if only a small sample of the population is able to reproduce, chances are there's going to be some alleles that are not going to be included in that small sample. So that's how genetic drift will tend to reduce genetic variation. In other words, reduce the number of alleles that are there at whatever locus you're looking at. So that's genetic drift. Now, one type of genetic drift, and here in the PowerPoint, they don't really mention that founder effect is actually a type of genetic drift. But anyway, founder effect is a type of genetic drift because it's a situation where only a few individuals become isolated from a larger population. And so basically only a few individuals, a very small number of individuals are founding a new population. So the founders of this new population are just a small number of individuals from a larger population. And so just like, so if there's just a small number of individuals that are forming this new population, chances are the allele frequencies in the small founder population are going to be very different from the larger parent population. And we could look back at this example of the flowers. Let's say you've got a population of flowers, and only 5 of those are the ones that are going to be the founders of the new population. Well, then the new population that's founded separate from this population is going to have a very different allele frequency compared to this population. And also, if just a small number of these were founders of a new population, just 2 of them that were red were founders of a new population, well, then that new population is going to be very different in its allele frequencies from this population or from this population. So that's what we mean by founder effect. And founder effect is just one specific type of genetic drift. And then now another thing, another phenomenon that's a type of genetic drift is called the bottleneck effect. And you can almost guess what this is. The bottleneck effect then occurs when for some reason, there's a drastic reduction in population size caused by whatever, sudden change in an environment, or caused by whatever it is. But for some reason, then there's a drastic reduction in population size, and so those that are left over, may not have allele frequencies that are reflective of the original population. In other words, the resulting gene pool may no longer be reflective of the original population's gene pool. And do you remember what a gene pool is? So what is a gene pool? A gene pool is all of the alleles of all the genes that are available, that exist in a certain population. If the population remains small, it may be further affected by a genetic drift then. And so here's a good example. So since we're talking about bottleneck effect, we're looking at little colored balls in a bottle. And let's say you've got a number of colored balls in a bottle, and this represents certain number of alleles at a certain locus in a population. So a few yellows and a large number of whites and blues. So let's say you dump out just a few of the balls at random from the bottle. So a bottlenecking event happens, and let's say you dump out only 6 balls, and lo and behold, you've got 5 blues and 1 white. So basically then, right at this point, this bottlenecking event basically eliminated the yellow alleles from the population that's left over. So understanding this bottleneck effect then can increase understanding of how human activity affects other species. Because a lot of times human activity can cause a bottleneck effect. For example, we can tie this into a cultural theme. Back in the 1870s, unfortunately, a lot of buffalo hunters went out and slaughtered almost all the buffalo, all the bison on the Great Plains. And so, there were only a few bison left, and so that was a big bottlenecking event. And so the bison today have a lot less genetic diversity than the original bison populations. So that's a link to culture here then, because it unfortunately had a very huge devastating effect on a number of tribes then that depended on the buffalo. And so, that's bottleneck effect then. Looking at another, here's a case study, the impact of genetic drift, or you could call it bottleneck effect as well, because bottleneck effect is a specific type of genetic drift. The impact of genetic drift on the greater prairie chicken. The loss of prairie habitat causes severe reduction, in other words, a bottleneck effect, in the population of the greater prairie chicken in Illinois. The surviving birds had low levels of genetic variation. Only about 50% of their eggs hatched. So here's the greater prairie chicken. And in 1820, there was a huge range for them in Illinois, but then because of converting it all to farmland, after the bottleneck effect, in the 1993 study, they found that there's very little greater prairie chicken left, and very little habitat for them left in the state. So that caused a great genetic bottleneck then. Here you can see the population size. Back in history, population size was very small. Number of alleles per locus was very small then, and the percentage of eggs that hatched. Then in 1990, percent of eggs hatched was very low. But the Kansas population had no bottleneck, and the Nebraska population had no bottleneck. So basically then, the researchers used DNA from museum specimens to compare genetic variation, museum specimens from a time before the genetic bottleneck. They used DNA from museum specimens to compare genetic variation in a population before and after the bottleneck. So that's how they were able to show that there was a loss of alleles, severe bottleneck, loss of alleles, and loss of genetic diversity. So these museum specimens are coming in really handy now that we have the DNA techniques to actually do all sorts of analysis on them. So the researchers, what they did then is they introduced greater prairie chickens from populations in other states, like Nebraska, like Kansas and Nebraska then. And so they introduced prairie chickens from other states. They interbred with the prairie chickens in Illinois, and they introduced new alleles into the population in Illinois, and that helped their genetic diversity, and it also helped increase the egg hatch rate to 90% then. So that's a good example of genetic bottleneck, and a good example of how we use our knowledge of genetics to help out a lot of natural populations. And they've done similar things with bison as well, also I'd like to mention. So genetic drift then, the effects of genetic drift. So genetic drift can be significant, especially in small populations. It can cause allele frequencies to change at random. It's a random change. And it can lead to a loss of genetic variation. We've seen that in a number of examples. And it can cause harmful alleles, possibly harmful alleles, to become fixed. What we mean by fixed is that they are the only allele that's there at a certain locus. So it's not fixed in the sense of being repaired, but it's fixed in the sense of being locked in and being permanently there, unchanging. So let's look at gene flow. Gene flow then is the movement of alleles from one population to another. Movement of alleles among populations from one population to another. So alleles can be transferred through the movement of fertile individuals, or sometimes alleles can be transferred just by movement of gametes. For example, if there's airborne pollen, that can actually blow from one area to another, and that pollen can take is basically gametes, then sperm cells, and it can take alleles from one population to another if it's carried by the wind for a long distance. Gene flow then tends to reduce variation among populations over time. And we'll look at some examples. It can affect adaptation to local environments. So, for example, variation in banding patterns in Lake Erie water snakes represents an adaptation to mainland and island habitats. So most snakes on the mainland, and so we're going to be seeing pictures of snakes here. So if you have a fear of snakes, I'm just letting you know now. Most snakes on the mainland are strongly banded. They have pretty good bands, but the island snakes are unbanded or intermediate, because unbanded snakes are better camouflaged in the island habitats. Excuse me. But migration of banded snakes from the mainland to the islands maintains alleles for banded populations in the island populations. So here's a banded one from the mainland, and then here's one from the island population, and you can see banding patterns. So on the Ohio mainland, there's much more banding than there is on the islands or on the Ontario mainland. So that's a good example of gene flow then. So gene flow can increase the fitness of a population. The fitness of a population is how well a population is adapted to the environment that it's in. So a good example of it then is the spread of alleles for resistance to insecticides. Insecticides then have become used to kill mosquitoes that carry West Nile virus and malaria. But mosquitoes have alleles that have evolved in some populations that confer insecticide resistance. And we saw that, a few slides ago when we talked about resistance in fruit flies. The flow of insecticide resistance alleles into a population can cause an increase in its fitness because they have a better resistance to insecticides. So gene flow then is an important agent of evolutionary change in modern human populations. And so evolution by natural selection is kind of involves both chance and also sorting of alleles. So new genetic variations can arise by chance, and then but beneficial alleles then are sorted and favored by natural selection. And only natural selection, though, consistently increases the frequencies of alleles that provide greater fitness or greater reproductive advantage. So let's take a little closer look then, and looking at natural selection. So it causes adaptive evolution. Remember that term, adaptive evolution, from a few slides ago. Adaptive evolution then by acting on an organism's phenotype. Of course, natural selection is acting on an organism's phenotype rather than genotype. And remember, phenotype is the trait or the characteristic that's caused by the genes that an organism has, or the alleles that an organism has. The phrase struggle for existence and survival of the fittest are a little bit misleading because they imply direct competition among the individuals. But usually reproductive success is generally more subtle, and it's not really direct competition. I don't know. In my opinion, there's a lot of direct competition among individuals in nature. But anyway, there's a lot of other factors as well. And now, when we look at relative fitness, when you say that one individual or one allele is more fit than another allele, it's all relative to each other. So relative fitness then is combination of the combination that an individual makes to the gene pool of the population, the gene pool of the next generation relative to contributions of other individuals. it's making a greater combination relative to what the others are contributing. And selection, then, natural selection favors certain genotypes by acting on the phenotypes of the individuals. Because natural selection can't act on a genotype, because a genotype is just what alleles are there. Natural selection can only act on the phenotype, because that's the actual traits, the actual characteristics that individuals have. And now there's actually, three different types of selection, or selection can act in three different types of ways. It can be directional selection, disruptive selection, or stabilizing selection. So it could, in directional selection, it could favor individuals at one extreme of the phenotype range, or it could favor individuals at both extremes of the phenotypic range, or it could favor the intermediate variants and not the extremes of phenotypes. So we'll look at examples though. So here's fur color in mice, and let's say there's a mouse population that has a pretty good variety of, a pretty good variation of fur color in that population of mice. Very light colored mice, varying all the way to very dark colored mice, and a lot of intermediates in between. And so here's the bell-shaped curve. And so most of the population actually has kind of an intermediate fur color, but there are a few at the extremes that have pretty dark color, and another few at the extremes that have pretty light color. Now, directional selection means that selection is acting against these at the extremes. So this directional selection is favoring those just at one end of the population. So it's favoring the darker ones. So it causes an evolved population to be, on average, darker than the original population. Then disruptive selection is favoring individuals at both extremes. Individuals that are light colored and also individuals that are dark colored. And so the ones towards the extremes increase in the population, and the ones towards the middle decrease. Then the opposite effect is stabilizing effect, where selection works against those that are extremely light colored, those that are extremely dark colored, and it favors those that are more intermediately colored. And so this is the new distribution. Then it becomes even more concentrated in the middle, in the intermediate forms. And so those are the three different types of directions for selection. So striking adaptations, in other words, very, very noticeable adaptations, very extreme adaptations, have arisen by natural selection. For example, certain octopuses and octopi can change color rapidly for camouflage. Or the jaws of snakes, and another snake picture coming up. The jaws of snakes allow them to swallow prey larger than their heads. A mammal can't do that. So the jaws of snakes are very specialized with a number of different joints and very flexible tendons then that are able to open very wide, and a snake can actually, like this snake swallowing this egg, it can actually swallow something bigger than its head. So natural selection then increases the frequencies of alleles that enhance survival and reproduction. So in other words, it increases those alleles that help the individual be more fit under certain environmental conditions. And adaptive evolution occurs as the degree to which species is well suited for life in its environment improves. So it occurs as it becomes more well suited for its environment. And because the environment can change, though, remember, environments can change. Because the environment can change, adaptive evolution is a continuous process, continuously reacting to changing conditions and things. So I'm going to I'm going to end part 2 of the lecture here. And in part 3 of the lecture, we're going to go on here and finish up the short section that's left for this chapter 23. So, contact me then. Remember, as always, look at your announcements every day, look at your emails every day for the class, and keep up with things. And I will be posting another lecture, part 3 of chapter 23, very soon. Thanks. Goodbye for now.